Hi, Assalamualaikum. Here we are from Class 6C3A Group 2 to present our Environmental Engineering Lab project. The group members are Amin, Rafiq, Dini and Akmal. The objectives of this project is to propose methods of measuring water quality samples, interpret the data and compare with the Malaysian standard as well as adequate treatment. We are taking the water quality index formula and calculation from the Department of Environment, and we are testing for 6 parameters that are dissolved oxygen, biochemical oxygen demand, chemical oxygen demand, ammoniacal nitrogen, suspended solids and pH. First, we are looking at the sample acquisition. We are taking from the tasik at section 7 Shah Alam from the inlet and outlet points of the lake. First, we are wearing personal protective equipment such as the lab coat and gloves before conducting experiment. For the dissolved oxygen experiment, we are taking 100 ml of sample and 200 ml of dilution water poured into a beaker and then into a glass jar. We then use a DO meter to take the dissolved oxygen reading. The next parameter we are testing is the biochemical oxygen demand. Based on the jars used in the dissolved oxygen experiment, we incubate them for 5 days to check for the dissolved oxygen after incubation. We are still using the DO meter and we are taking the dissolved oxygen value after 5 days of incubation and perform calculations to obtain the biochemical oxygen demand of the samples.
Okay, next parameter is about chemical oxygen demand, COD. First, we are using apparatus HRVL, LRVL, COD reactor, pipette and spectrophotometer. First, you need to prepare one HR and one LRVL for each sample. These RVL are used for inlet sample, outlet sample and blank sample. A pipette will use to insert 2 ml of each sample into their respective vials. The vial will shaking to evenly mix the solution. All the vial will place into the preheat COD reactor for 2 hours. And after that 2 hours, the vial will left to cool in room temperature. Spectrophotometer was then used to measure the COD reading by setting the blank vial to 0Z and then setting the HR and LR vial for each sample respectively. The reading for each sample 1 and 2 for HR and LR will recorded.
Next, we'll be testing for ammoniacal nitrogen. We'll be using a measuring cylinder, beaker, sample cell, pipette, mineral stabilizer, polyvinyl alcohol, Nestle reagent as well as a spectrophotometer. First, 25 ml of sample was measured in a beaker, which was then poured in a measuring cylinder. Then, 3 drops of mineral stabilizer, which equivalents to 1 ml, was added to all 3 samples. Next, we will have 3 drops of polyvinyl alcohol as well as 1 ml of Nestle reagent, which was added using an auto pipette. The solution was then mixed and left for 1 minute for the sample and solution to dissolve. 10 ml of each sample was inserted into the sample cell for us to take the reading using a spectrophotometer. The blank cell using dilution water was used to zero-rise the reading of the spectrophotometer. Each cell was wiped using a tissue to obtain accurate reading inside the spectrophotometer. Next, we'll be testing for the suspended solids in the water sample. We'll be using a metal dish, weighing balance, oven, furnace, glove tongue, desiccator as well as a vacuum pump. First, we measure the empty dishes. Then, we will pour 20 ml of each sample into the metal dish. The dishes were then placed in an oven which has an average of 105 degrees Celsius for 6 hours. After 6 hours, the samples were placed in a desiccator and the vacuum pump was used to cool down the metal dishes inside the desiccator. After 2 minutes, the desiccator was turned off and we weigh the metal dishes using the weighing balance. After obtaining the weight, we will use tongs to place the metal dishes inside a furnace with an average of 550 degrees Celsius for about 30 minutes. After 30 minutes, the metal dishes will place again in a desiccator for 5 minutes to cool them down, and they will weigh and take the reading.
Next experiment is a pH reading. The apparatus is a pH meter and beakers. First is a need of 100 ml for each sample to spray in the beakers and start the pH meter. So that record a pH value on the reading has a stabilizer. The next experiment is about E. coli. The apparatus for E. coli is a standard coliform sample, E. coli sample tray, colilert, sealer mood, sealer machine, and E. coli apparatus glass jar. Okay, next is a UV light and E. coli MPN table. Okay, first is need a measure 110 ml of each sample using a measuring cylinder. The sample was poured into glass jar. Insert the one portion of colilert into the glass jar. Put on the cap and shake the glass jar until the colilert fully dissolved. Pour the sample solution into an E. coli sample tray. Keep the pouring angle at about 45 degrees to minimize air bubbles. Place the E. coli sample tray into the sealer mold and seal the sample tray with the sealer machine. This experiment need to repeat 2 times because have 2 samples. After that, after seal the sample, need to put the sample back into the E. coli apparatus and leave for 24 hours. After 24 hours, the E. coli sample tray was taken out. A patient of a coliform is verified by comparing the yellowness of sample into the E. coli sample tray with the standard coliform sample. Check the presence of E. coli by using a UV light. The number of luminous square under the UV light was counted and the E. coli data was taken by referring the number of box with the respective value in the E. coli MPN table.
After conducting all the experiments for the WQI parameters, we will check the results and analyze them according to the water quality index. We will do calculations for dissolved oxygen, biochemical oxygen demand, chemical oxygen demand, ammoniacal nitrogen, suspended solids, as well as pH, and we will calculate all of the given sub-indexes in the WQI formulae. According to the formula, we know that the lake is classified under class 4 due to its WQI range being between 31 to 59. These are the Department of Environment parameter set that we use for checking the WQI. Thanks for watching.